Hello, good morning everyone. Today's topic is bacterial structure and genetics. So bacteria is a heterogeneous group of unicellular organism with about 1 to 8 micrometer in diameter. Bacteria comes under prokaryotic division, has primitive nucleus with one chromosome. They don't have any kind of nuclear membrane, no mitochondria, no sterols, and no ribosome in it, so they are unable to produce their own protein. A bacteria has extra chromosomal structure called plasmid. The structure of bacteria shapes. Bacteria may be spherical or oval, cocci, rods, bacilli, or very short bacilli, that is cocobacilli. A tapered end called fusiform bacteria, a club-shaped and curved bacteria called vibrio, helical and spiral bacteria called spirochetes. Here are different morphologies of bacteria you can see in the diagram. First one is pneumococci, streptococci, pseudomonas, salmonella typhi, staphylococci, mycobacterium, clostridium tetani, and treponema and leptospira, which comes under spheres, rods, and spirals. Arrangements of bacteria is in amongst cocci. When bacteria are present in pairs called diplococci, when they are present in chains called streptococci, when they are present in cluster called staphylococci, when they are present in group of 4 called tetrad, or when they are present in palisades called corynebacterium. Here is the structure of bacteria or cell wall appendages. The cell wall of bacteria are with rigid cell wall surrounded cytoplasmic membrane and internal structure. A function of the cell wall is to maintain rigidity, shape of bacteria, protection. They are porous and permeable to particle molecules. They help in cell division and antigenic determinants. The structure of cell wall: two groups of cell wall depending on the reaction to gram stain. First one is gram positive bacteria and second is gram negative bacteria. Chemical structure of the cell wall: it's composed of peptidoglycan layer, which is rigid part, mucopeptide composed of alternating strands of NAM and NAC, that is N-acetylmuramic acid and N-acetyl glucoxime, linked with the peptide subunits. There is a structure of bacterial cell on molecular basis. First one is that is an N-acetyl glucoxime and N-acetylmuramic acid, which combine to form peptidoglycan layer. Here is the procedure of gram staining, which differentiate gram positive and gram negative bacteria into two different groups. The first step of this procedure is to take a slide or wipe bottom of the biofilm slide clean. Second is clean top edges of the slide about 2 mm. The third step is to build a ridge of petroleum jelly on the top and bottom of the cover slip. Fourth step is cover slip with petroleum jelly. Fifth is biofilm on slide with cover slip. Sixth is add crystal violet for about 30 seconds. After that, wash with water. Add gram iodine for 1 to 5 minutes. Decolorize with alcohol. Again wash with water. Add safranin, that is counter stain, for 30 seconds. After that, wash with water and observe it under 100x by using oil immersion. The gram positive bacteria are purple in color and gram negative bacteria are pink in color. Gram negative cell wall, which having a thin peptidoglycan layer. Outer membrane of gram negative contains specific protein called porins, which are important in the transport of hydrophilic molecules. Lipopolysaccharide and lipids are produced by the gram negative bacteria called endotoxins. Gram positive cell wall: peptidoglycan layer of gram positive bacteria is thicker as compared to gram negative, closely associated with cytoplasmic membrane, composed of teichoic acid, which anchors cell wall to cell membrane, epithelial cells adhesion. Antigens are polysaccharides. Here is the structure of a gram-positive bacterial cell wall. The external structure of a bacterial cell, they compose or protrudes from the cell into the environment. Like they have flagella, pili, fimbriae, or capsule outside of the cell wall. First one is flagella, that is helical filament composed of protein called flagellin, found in both gram-positive and gram-negative bacteria. There are of different types, that is peritrichous flagella, monotrichous flagella, and Lophotrichous flagella. Here is the diagrammatical representation of these flagellas. First one is monotrichous, having single flagella. There is Lophotrichous, that is having multiple flagella, and peritrichous, so it's having all flagellas all over the bacterial cell. Pili are also the outside protruding structure of the bacterial cell, which is a fine short filament extruding from cytoplasmic membrane found on the surface of the many gram-negative and gram-positive bacteria. The pili are composed of a protein called pilin, having two classes. Common pili, called fimbriae, cover the surface responsible for addition and colonization. The second one is sex pili, in some bacteria only responsible for conjugation. There is another one structure which covers the bacterial cell called capsule, which is amorphous material surrounds bacteria, usually composed of polysaccharides, occasionally proteins. Function: inhibit phagocytosis, act as virulence factor in some bacteria by assessing attachment to the surface. There is a cytoplasmic membrane, which is called cell membrane. It's outside of the cell, which is double layer and structure composed of phospholipid and proteins. Act as semi-permeable membrane for the passive decision. Site of numerous enzymes which involve in active transport of nutrient and various metabolic processes.
Buy and sell share